What can I do for you?
How does the process of glycolysis occur? Firstly, it starts with the molecule of glucose that enter the cytosol. This reaction takes place from the conversion of glucose into glucose 6-phosphate. The enzyme that catalyzes this reaction is called as hexokinase. The hexo means denoting the 6-carbon compound of glucose, while the kinase is the group of enzymes that catalyze phosphorylation reactions. The enzyme of hexokinase transfer a molecule of phosphate from ATP to glucose and convert glucose into glucose 6-phosphate. In this reaction, ATP is converted into a molecule of ADP. Hence, it utilizes one molecule of ATP. Next, the glucose 6-phosphate is converted into fructose 6-phosphate by the enzyme called isomerase. This is the simple rearrangement reactions. In the next step, the fructose 6-phosphate is converted into fructose 1,6-biphosphate. That is the addition of another phosphate group at the first carbon of the fructose ring. This reaction is catalyzed by the enzyme that known as phosphofructokinase. Another kinase enzyme is transfer a group of phosphate from ATP to fructose 6-phosphate, and again ATP is converted into ADP. Hence, another ATP is utilized in this reaction. Next, the fructose 1,6-biphosphate is split into two, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These are 3-carbon compound, and the enzyme that catalyzes the reactions is aldolase. All the reactions are composed the first phase of the glycolysis, and as you can see, the energy is actually utilized in this process in the form of 2 ATP. The two compound, which is glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, that actually isomers and can be converting into each other by the enzyme called as isomerase. The glyceraldehyde 3-phosphate is further utilized in this process of glycolysis. That equilibrium of these isomerization reactions is always to a glyceraldehyde 3-phosphate, also keeping in mind that every molecule of glucose split into 2 molecules. So, every reactions of the second phase happen twice and produce twice products. In the next reaction, the molecule of glyceraldehyde 3-phosphate is converted into 1,3-biphosphogycerate. This involves the addition of phosphate group at the first positions of the phosphoglycerate. The enzyme that catalyzes these reactions is called as glyceraldehyde 3-phosphate dehydrogenase, and the coenzyme of NAD is reduced to NADH, which is further utilized in the electron transport chains to produce more energy. Also, this reaction utilizes a molecule of inorganic phosphate. Then, the 1,3-biphosphoglycerate is then converted into 3-phosphoglycerate by the enzyme of phosphoglycerate kinase. This kinase enzyme transfers the phosphate group adipose positions of phosphoglycerate into adenosine diphosphate, and this process proves that a molecule of ATP is generated. Next, the 3-phosphoglycerate is then converted into 2-phosphoglycerate. This is the simple rearrangement reactions that occur with the enzyme phosphoglycerate mutase. Next, the 2-phosphoglycerate is converted into phosphoenol pyruvate, where involving the enzyme of enolase and magnesium ion. Next, the phosphoenol pyruvate is converted into pyruvate by the enzyme of pyruvate kinase. The phosphate group of the phosphoenol pyruvate is transferred to ADP and converted to ATP. This reaction is composed of the second phase of glycolysis, and as you can see, the energy is produced in terms of ATP. Then, The net reactions of glycolysis at the splitting molecule make a better understanding on what are the actually product of glycolysis, which are 2-ADP, 2-PI, which is the phosphate inorganic, 2-NAD+, and 2-pyruvate, 2-ATP, and 2-NADH. So that's all from me.
Assalamualaikum warahmatullahi ta'ala wabarakatuh. I am going to share my slide in order to make us more easier to remember the process in Krebs cycle. I hope we can learn it together, and also I hope you guys enjoy the slide. Krebs cycle, also commonly known as citric acid cycle or tricarboxylic acid cycle (TCA). Where does the Krebs cycle take place in the cell? Basically, the Krebs cycle takes place in mitochondria matrix. In Krebs cycle, it's involved in several steps. Before I explain the process in details, let's take a look at an idea of Krebs cycle. In the first step of Krebs cycle, acetyl coenzyme combine with oxaloacetate to form citrate. After that, the carbon released from the citrate will produce NADH. As it has 6 molecules, so the remaining 4 carbon molecules will undergo to produce ATP or GTP. This will affect the electron carrier FADH to reduce to FADH2, and once again NADH will be produced. As overall, to be short, Krebs cycle is when carbon dioxide that have been released produce 3 NADH, 1 FADH2, 1 ATP or GTP. The figure shows how the Krebs cycle occurs in mitochondria. The Krebs cycle, also known as amphibolic, in that it is both catabolic, which is break down molecules, and anabolic, builds the molecules. Here it is the figure of the Krebs cycle. By looking at it, it seems that really difficult for us to memorize all the molecules, but don't worry, we will learn it step by step. Reactions involved in the Krebs cycle. Now I will explain it in more detail. In Krebs cycle, there are 8 steps that I need to memorize and understand. As we know, glycolysis and Krebs cycle are linked to each other. This is because the end product, which is pyruvate in glycolysis, will split into 2 carbon and 2 acetyl group. The carbon will diffuse out of the cell, and the acetyl group will combine with coenzyme to form acetyl coenzyme. This product will further be used in Krebs cycle. The first step in Krebs cycle is formation of citrate. Acetyl coenzyme A will combine with oxaloacetate to form citrate. Bear in mind, the final reaction in this cycle is involve the regeneration of oxaloacetate. The enzyme that is involved known as citrate synthase. Second step is formation of isocitrate. The citrate will be converted into its isomer, which is known as isocitrate. The process involves dehydration followed by hydration of water by the enzyme aconitase. After that, the third step is oxidation of isocitrate and formation of carbon dioxide. In this step, it is the first redox reactions that occur in the cycle. The reaction is catalyzed by isocitrate dehydrogenase at where first the isocitrate is oxidized into oxalosuccinate. Second, NAD+ will be reduced to form NADH and the oxalosuccinate will undergo decarboxylation to form 5 carbon species which known as alpha ketoglutarate. In this step, the first molecule of carbon dioxide and NADH are produced in the cycle. Next, we are going into the fourth step, which is oxidations of alpha ketoglutarate and formation of carbon dioxide. The alpha ketoglutarate is oxidized, reduce NAD+ to NADH, coenzyme A, and then will forming the unstable compound known as succinyl coenzyme A. The enzyme that being used in this step is known as alpha ketoglutarate dehydrogenase. So therefore, there are 3 product forms in this cycle, which are carbon dioxide, NADH, and the 4 carbon species known as succinyl coenzyme A. Without wasting any time, the fifth step is thioester bond cleavage in succinyl coenzyme A phosphorylations of GDP. ATP or similar GDP or similar to ATP and free phosphate group will react to succinyl coenzyme A. The energy release will combine the ATP and free phosphate group to form GTP, which is guanosine triphosphate. The succinyl coenzyme A will be converted into succinate. For the step 6 is oxidations of succinate. This is the third redox reactions that occur in the cycle. Succinate dehydrogenated by FAD catalyzed by succinate dehydrogenase to produce fumarate at 4 carbon species with trans double bond. The FAD will be reduced to form FADH2. For the 7th step is hydrations of fumarate. The enzyme fumarase catalyzes the additions of water to the double bond fumarate and produce malate molecules. And for the last step is oxidations of malate to regenerate oxaloacetate. This is the 4th redox reactions of the cycle, at where NAD+ will react with malate produce NADH. It will be catalyzed by using malate dehydrogenase. The product, which is oxaloacetate, will combine with another acetyl coenzyme A and the cycle repeated again. That's all from me. Thank you.
Alright, so for the next one will be the electron transport system, or we can call it as electron transport chain, simply ETC. ETC takes place in the membranes embedded in the mitochondrion known as the cristae. ETC specifically occurs in the intermembrane space and the matrix of the mitochondrion. Two reducing equivalents, namely NADH and FADH2, resulted from the Krebs cycle contains electrons that will be donated or passed down to a set of enzyme complexes where the ETC process will occur. In the matrix, they are packed with high concentrations of hydrogen ions, and they are needed to be transported to the intermembrane space. When the hydrogen ions were continuously pumped into the intermembrane space will eventually generate a higher concentration of hydrogen ions than the matrix. Thus, this will lead to the electrochemical gradient, where the hydrogen ions are inhibited to flow back to the matrix due to the inner mitochondrial membrane, which are non-permeable to ions. Thus, oxidative phosphorylation takes place, where the F1FO ATP synthase, or simply called complex 5, will be a good help in transporting the hydrogen ions back to the matrix, and this movement is called the chemiosmotic movement. The energy liberated from the flow of hydrogen ions back to the matrix are used to phosphorylate ADP to ATP. Therefore, ATP is synthesized. So, for the electron transport chain, it involves 4 enzyme complexes and 2 prosthetic groups, namely coenzyme Q and cytochrome C, that is responsible in the chains of the electrons. The enzyme complexes were numbered in Roman numerals from 1 to 4 respectively. The ETC begins in complex 1, named NADH CoQ oxidoreductive complex. NADH was oxidized to NAD+ and H+, which donates 2 electrons to flavin mononucleotide, which will then be further reduced in giving another 2 electrons to iron sulfate. Then, ubiquinol is further reduced to ubiquinone by receiving a pair of electrons from iron sulfate. Simultaneously, energy is liberated from the electron movement to pump hydrogen ions into the intermembrane space. The electrons were transported to complex 3 via coenzyme Q. Meanwhile, in complex 2, named succinate dehydrogenase complex, succinate is oxidized to fumarate, where this is a step that occurs in Krebs cycles that loses electrons to reduce FAD to FADH2. Then, the FADH2 will lose 2 electrons to iron sulfate, which the electrons will be transported by the coenzyme Q to complex 3. Next is the complex 3, or we can call it as cytochrome C reductase, that contains 3 types of cytochromes, namely cytochrome B, cytochrome C1, and cytochrome C. The main function of this complex is to transport the electrons received from the coenzyme Q from both complex 1 and complex 2 to complex 4 via cytochrome C. Bear in mind that the electrons were transported one at a time to complex 4. Meanwhile, energy resulting from the electron chains will also pumps the hydrogen ions from the matrix to intermembrane space. Last but not least will be the complex 4, or we can call it as cytochrome C oxidase, that will oxidize cytochrome C in order to receive the electrons that will be used in reducing oxygen molecules to form water molecules. Similarly to the other complexes, energy was harnessed from the electron chains or the movements to pump hydrogen ions into the intermembrane space. Since oxygen is the final electron acceptor, the overall process is called aerobic respiration. So that ends the electron transport chain.